Brad and the others for inviting me. I have to say this project is just starting up in 2011, so we have lots of promise, but I'm very excited and enthusiastic about this project, as I'll hopefully convey. But first, I just want to bring up a couple of things regarding some other bioinformatics projects that are continuing after some Genome Canada support from involving other projects, just to bring up the fact that there's a lot of Genome Canada projects that can get things going, but then the work can't continue. So, for example, excuse me, there's an analysis of tuberculosis genomes for a TB outbreak that was done as part of some Genome BC funding, where we are now continuing with further examinations of some BC isolates, but also expanding to some analyses in India, where, of course, TB is a significant and growing problem, particularly with drug-resistant TB. And then also with a SOF project where we're doing bacterial transcriptomics. We're now about to finish up a project involving doing RNA-seq analysis of bacterial genome in a level of detail that, in my mind is unprecedented, and I'm really enthusiastic about releasing this data because it really reveals the complexity of RNA cleavage events that are occurring, as well as traditional transcription. And this is actually relevant also for eukaryotic genomes, and I want to impress upon you, if you're doing any transcriptomics, to realize there is a lot of cleavage and other events that you may not be capturing through your RNA-seq traditional analyses. And finally, through this couple of pathogenomics projects I've been involved in, we're continuing with some network-based analyses of gene expression responses using RNA-DB bioinformatics platform, and continuing to expand this database and incorporate new pathway analyses. And I just thought I'd mention this one in particular. This is something we're just writing up for publication right now that may be relevant to some of you who are doing any kind of pathway analysis. We'll also use this with this Metagenomics project, in that what we're trying to do is identify pathway signatures with this tool we call Sigora for signature pathway overrepresentation analysis. And the concept is that you may find a bunch of genes associated with pathways, but some of those genes, like say G5 here, is not so significant, telling you any useful information about, because if it gets upregulated, it sort of is involved in pathway 1, pathway 2, and pathway 3. But pathway gene 2, if that gets upregulated, that's really likely indicating that something is happening with pathway 3, because it's only associated with that, or these gene pairs, G3 and G4, are really associated with pathway 2. And in short, we find by looking at the signatures of pathways, or genes that are uniquely associated with a pathway, we can get a much better signal in terms of what pathways are really being affected in analysis, and not get biased by these sort of generalist genes that will make all sorts of common pathways show up all the time in analysis. But I'm going to move on to talk about Watershed Metagenomics. So first, just to make sure everybody's very clear, metagenomics is a study of all genetic material recovered from an environment and helps describe an environment's microbiome. And also, just to define, a watershed is an area that drains all precipitation received into a river or set of rivers, and is obviously affected by all that the water comes in contact with from any pollution that may be introduced into the water through what kind of soils or whatever the water may interact with. I bring this up because I was amazed. I gave a talk on this project at Harvard a couple of months ago, and how nobody in the audience seemed to know where their water was coming from. So just to make sure you guys all know where your water is coming from, Metro Vancouver, it all comes from this Capilano watershed, Seymour watershed, and Coquitlam watershed. Granted, in Boston, it's a little more complicated where the water's coming from, but we have the benefit of these are, excuse me, 100% protected watersheds, whereas in many communities, watersheds are not as protected. So we have really great water sources here. But obviously, from both an environmental standpoint and public health perspective, we really care about trying to protect our water quality. And certainly, this is the main focus of this project. The reason we care about Watershed Metagenomics is also that we really want to move from this current emphasis of testing water quality at the tap and moving more towards testing at the source in the watersheds, identifying pollution earlier in the process. And also another issue that's significant is the fecal coliform test, a test that's very commonly used for tracking water quality, is really inaccurate, has many false negatives in not detecting non-coliform pathogens, and then a lot of false positives in that not all pathogens, such as protozoans, are coliform. So we really need better water quality tests. We need to identify if fecal pollution is present, and also, what is the source of this pollution, if we can try to remediate earlier and identify source of pollution earlier. We also need better indicators, both bacterial, viral, and potentially protozoan markers. And really, to do this in environmental surveys, what's needed to find these indicators, and metagenomics really is the tool that can do this survey. So hence the Applied Metagenomics of the Watershed Microbiome project started last year, that is really got two major phases involving doing metagenomic survey of the watersheds, targeted watersheds, I'll describe, and then developing within the 3-year project PCR-based quality and attribution, or identifying the pollution source tests. And I'm really enthusiastic about the potential of this project because it also has a very strong integrated GELS project, understanding stakeholder needs, understanding the regulatory framework that is very directed to figuring out how we can ensure that this really gets implemented as quickly and efficiently as possible. And this project is being led by Patrick Tang and Judith Eisen-Renton, who are here involved in this, who are leading this project, and they basically at the BCCDC have the mechanism to basically directly translate this, because unlike in many provinces where Environment Canada does water quality testing, in BC our water quality testing is done at the BCCDC, and they have a lot of experience with developing PCR-based tests, basically. So literally we can come up, do this metagenomic survey, come up with these tests, and implement those tests within 3 years. And this study, I'm just going to show one slide before I get into the bioinformatics about just the overall study design. So the idea is to look at a control or a protected watershed that's pristine, and we're actually using the Sook watershed in Victoria, so I'll know what Victoria's drinking water is like. And then we're looking at 3 watersheds, human impacted, agriculturally impacted, and mixed impacted, where we're collecting the water over 1 year with metadata, microbiological, chemistry, physical data, and then filtering the water for protists, bacteria, and viruses. Unlike some projects that have done metagenomics focusing, say, just on bacterial 16S sequences, we're doing the amplicon sequencing, or these targeted like 16S sequencing, but also viral and protist sequencing, so we're going to capture more microbes, and also we're doing Lumina shotgun sequencing of the bacteria and viral enriched samples in order to get a really better holistic view of what's happening in the microbiome for these watersheds. For the bioinformatics analysis, there's some huge challenges as we essentially target 3 main areas. Really what you're interested in is who is there, what are the different members of the community doing, and how those communities are different. For who is there, we're really just looking at taxonomical binning, as we call it, looking at the phylogenetic or taxonomical composition of the sample. We can do that amplicon-based or shotgun-based using sequence similarity or sequence composition. And then for what the different members are doing, we really focus on sort of more functional binning of gene functions, and the idea is the communities, as they change, there's different proportions of organisms that will have different proportions of genes associated with certain metabolic properties, and so you can see these communities change in terms of literally what the gene function, proportions of genes of certain gene functions that will change over time. And then doing community comparisons based on these profiles, and really the main goal is to lead to these practical applications of biomarkers, getting away from the fecal coliform test, moving towards a really more accurate test that can also enable us to do source tracking. So The challenges for this are significant, as I'll illustrate. There's notable hardware requirements, as is with many bioinformatics genomics associated projects with genomics. But in this case, certainly we've got the benefit of Westgrid and resources that have already been set up thanks to previous Genome Canada projects that really allowed this to move forward quickly. Another challenge that is happening a lot in many bioinformatics areas is sort of lots of new software methods coming out all the time, and it's sort of hard to evaluate them because a lot of them, for example, in metagenomics, there was a dozen new notable methods published in 2011, which are all coming out at the same time, so they've never been compared against each other. And so we really have to have set up a procedure to be able to compare these methods very rapidly as they get released, figure out what methods that we should best be implementing. So one of the things we've been doing, just to highlight, is using a tool called Metasim that allows us to simulate sequence reads for metagenomes, and then we can evaluate methods based on these simulated reads. So this Metasim program, which can actually allow you to just make collections of synthetic reads from any genome theoretically, literally just chops up you pick bacterial genomes that you want to have in your metagenome, or you can pick just one genome if you just want to do one genome, and it literally slices it up into whatever size reads, paired end reads that you want, and it can produce even evolved sequences with mutating them, and you can simulate different sequencing technologies. And then the idea is that we're going to be evaluating all of the methods that come out and set up a pipeline for being able to evaluate as new methods inevitably come out over the coming year. Both with their data set, they have a data set they're trying to use as sort of a set that you should analyze all methods against as sort of a benchmark, and then we're going to develop some sets based on species found in water based on our pilot data to allow us to look at species that are more relevant in our case, and then real data sets where we won't know the truth, but at least we can compare differences across these programs. So I'll use this as a little bit of a cautionary note in doing bioinformatics. One of the things you really have to be careful about is to evaluate methods very well and see where the problems are. Just doing going into a web page and clicking and getting a result can be very misleading sometimes. So here's an example of a test data set of 11 species where you can see the percentage of reads of each species that's been basically a simulated reads, not evolved, so they're 100% identical to sequences in GenBank. So of course, metagenomics classifiers should do pretty darn well with this versus a real data set where you get a lot of species in there that have never been sequenced before. But this is the kind of data you get. So here's true data. This is using default settings for MGRAST and Megan4, two popular metagenomics programs. Granted, there's another one we think is better that we haven't tested yet. But basically, you'll see that you get actually many species sort of being correctly identified. You get all of this stuff that shows up. For example, there's Streptomyces silicolor, which has hits occurring to a whole bunch of different Streptomyces, and it's literally just because you get sequence reads that just across species are basically 100% identical. And so just if the BLAST analysis sees another species first, it will just potentially mark that species artificially as the true species, even though these programs try to factor in when they're seeing a lot of reads. So there's definitely a problem in the end with the species level classification, with species abundance and false positives being a problem, because really we should be seeing those 11 species come back out. If you actually look at general level classification, you still have a problem with a lot of these guys. Like, for example, Anaemia is very similar to nostoc. So you see some reads that get classified that were truly Anaemia as nostoc. But you'll see that certainly at the general level, you get pretty darn good classification, but you definitely have a problem with these extra genera. And it's amazing how many genera can actually get classified. But in short, you have to be really careful in doing bioinformatics analysis. You do a lot of these tests, and in short, that's what we're focusing on right now at the start of the project, so we can be ready once the data comes in to hit the ground running with really careful, conservative bioinformatics analyses.